Welcome to Brain Ponderings, your window into the world of neuroscience research with experts at the forefront of their particular subdisciplines in brain research and related disciplines related to brain search research. So today it's my great pleasure to welcome Jonathan Long. He's Associate Professor of Pathology and an Institute Scholar of Stanford Chemistry, Engineering, and Medicine for Human Health at Stanford University. He also has appointments in neuroscience, cardiovascular institutes, and other institutes at Stanford University. He has some very exciting findings, very recent findings, where he's identified a couple biochemicals that change when you exercise or when you fast, that his evidence is they have really interesting and potentially important effects on whole body metabolism and even neuroplasticity, for example, learning and memory. So welcome, Jonathan.
Thanks. It's great to be here.
Okay, so before you get into these two molecules that you can focus on, which are very exciting to me or anyone who's following the fields of exercise, fasting, longevity, even, general health, metabolic health, and brain health will be interested in these and may not know about them. In fact, I think probably don't know about them. Um, so you went to Columbia University?
Yeah, I'm a Columbia alum at a time when Columbia was not a controversial university.
But it still shouldn't be, right?
It still should not be. No, I was there as an undergrad. I graduated with a degree in biochemistry.
Yeah, and I went back on PubMed and I went back to your time at Columbia, and you must have been working in some labs when you were an undergraduate because I see you're an author on a few papers.
I was in New York all 4 years. I stayed in New York during the summers. Obviously, New York is a very thriving scientific city. There's Columbia on the Upper West Side. There's the Upper East Side with Cornell and Rockefeller and Sloan Kettering. And of course, there's
Einstein.
Einstein. There's NYU.
Yeah.
So I was up at Columbia, during the academic year, and then during the summers, I'd go down to Sloan Kettering and that tri-institutional area to do a summer research internship. And so that's where I cut my teeth in scientific research at those two places.
Yeah, that's you were completely immersed in science. As everyone who's in science from undergraduate on up knows, it's like at least 60 hours a week minimum.
Well, also, I think at that time, and still now, New York is such a fun city to be in. And so, over the summers, I thought, well, why leave? I can just stay here. And so that's
Yeah. Yeah. Okay, and then you went to the West Coast from there.
yeah, first time ever setting foot in California. I was 22 years old, came with my two suitcases, landed in San Diego. I started grad school there at a relatively small program, even now, Scripps Research Institute. And then I got my PhD there with a wonderful advisor, Ben Cravat, and my PhD was in chemistry.
Yeah, and so the San Diego, there's a lot. It's a really hot place for science in general, biomedical sciences. There's a bunch of biotech companies. There's UC San Diego, which has the second best neuroscience program after Johns Hopkins. No,
Yeah,
I'm
I
kidding.
think
I'm kidding.
I think I mean, no, San Diego
Yeah.
and especially Scripps was really great. When I was an undergrad, even though my degree was in chemistry, actually most of my research, especially towards my later years, was focused in chemistry, synthetic chemistry.
Ah,
I've always been, sort of very interested in this interaction and intersection of small molecules and physiology. Actually, at the time, the primary focus of my thesis lab was sort of on psychoactive alkaloids. And so it was sort of a very interesting time where you could see where I had all this training in research and training in chemistry, but you could really see that the power here was towards human health, towards sort of molecular mechanisms of physiology. And so part of the reason that I chose to go to San Diego at that time was because my advisor, Ben, was sort of one of the few people in the still nascent field of chemical biology
Uh,
who was really, in my opinion, truly bridging chemistry and biology. You he had organic chemistry fume hoods in his lab. He was doing, making molecules. That's something, of course, all the chemists like. But he was the only person who not only had that, but also, for example, had knockout mouse models and like a whole suite of mass spec and proteomics equipment. And so I thought that that intersection was really fascinating. I mean, the whole field of chemical biology was still sort of developing and trying to define itself. And I thought, well, if I'm really going to have a chance to translate some of the chemical knowledge into uh, an understanding or things that really matter for human health, Ben's lab was really, in my opinion, sort of the place to be to learn sort of the intersection of all those fields. And so that's why I went, and that was sort of the original motivation.
and you, from what I can tell, what was really maybe a little bit unusual and good is that you had an interest in different types of molecules and metabolites, lipid uh, metabolites, for example, right? You worked on endocannabinoids, was that
Yeah, in
right?
my PhD, exactly.
And these are lipid-like molecules, and then maybe this makes a lot of sense now, how you ended up looking at the
So
at a conjugate of a lipid and an amino acid.
yeah, so maybe just to take a step back first, in my PhD, my thesis work was focused on the endocannabinoid system. Most people are probably familiar with cannabis. I
Right.
mean, this is the active component of marijuana. So when you smoke pot, you get high, and that's because there's a small molecule in there, tetrahydrocannabinol, which activates these receptors in your brain called the cannabinoid receptors. But of course, the cannabinoid receptors are not in your brain so that you can get high. That whole cannabinoid system is very important for many, many different behavioral and physiologic processes, including pain and anxiety and locomotion and even body weight control. And so my thesis was primarily focused on the natural ligands of the cannabinoid receptors, these so-called endocannabinoid molecules. So we make our own THC-like molecules. And we were studying their metabolism and their biosynthesis and their catabolism and trying to develop selective chemical tools that could perturb their levels, for example, in a mouse, to understand what the natural system was doing. And so it was from that I really, so sort of coming from more of a synthetic chemistry background, that was sort of my entry point to understand, well, how do you use chemistry to probe complex biological systems? In this case, one of the most complex biological systems, an intact organism. And, uh, and and it happened to be in this very interesting area of lipid neurotransmission and neuroscience. It happened to be in a very interesting set of phenotypes related to behavior and cognition and feeding control. And and so that's sort of what I was doing in my
Uh,
PhD. And it was from there that I really then
uh,
developed an interest in not only understanding how molecules control physiology, but then in trying to understand, well, how did people actually discover this stuff to begin with? I had always thought that, even though I was studying the endocannabinoids and I was studying the cannabinoid receptors and I was studying all of this downstream physiology associated with those molecules, what I had always admired, even at that time, were the papers describing the original cloning of the cannabinoid receptors, the original discovery using radio-labeled ligands of the endogenous ligands to the cannabinoid receptors, the endogenous agonists, our homebrew THC in our brains. And I remember reading those papers and just thinking, these were the seminal discoveries that catalyzed this entire field. What I really need to learn as sort of my next step is how to make such discoveries, how to connect molecules to physiology, sort of de novo, not just building on what other people had done, but really doing that from scratch. And so that's when I went to Bruce's lab, Bruce Spiegelman, at the Dana-Farber in Harvard for my postdoc, because He had a long history of discovering molecules to physiology. And he also happened to be working in an area that I had a personal interest in, which is metabolism, and sort of energy balance and energy homeostasis.
Okay, and there, yeah. So there's, yeah, his labs had some really interesting discoveries, and that. So that's where he got it interested specifically in exercise. I mean, he'd done some work. There's this. I don't know if it was who coined the term Exerkines, but these molecules that are released in the blood when you exercise and they do something.
Right.
But
Well, the original well, maybe let me just say yes. So Bruce, of course, he's had a very long and prolific history in molecular metabolism, very sort of storied history, discovery of PPAR gamma, PGC1 alpha, many very important and interesting molecules.
yeah,
It was in his lab that I first got introduced to the idea that you could actually study exercise through a molecular lens. Before this, I mean, I played sports, I sort of I still work out. I get, your recommended dose of physical activity that the CDC tells you to get, the 150 minutes a week. But I never thought of exercise as something you could study. It's just too complicated, right? There's like so many different things going on. People can't even agree on what exercise is. It was in Bruce's lab that I understood that not only could you study it, but you could apply the same level of scientific rigor to the study of exercise as you would any other observation. That this is fundamentally a molecular process that you can look at this complex physiologic system through the lens of molecules and then decipher how individual molecules contribute to the complex phenotypes. And so Bruce's lab at that time was sort of working on this idea of myokines, that when you're exercising, that muscle is releasing certain factors that then are sort of circulating endocrine hormone-like molecules that mediate tissue crosstalk and at least mediate potentially some of the salutary effects of physical activity. I got introduced to the idea of myokines. Of course, Bruce was doing it through the molecules that his lab had discovered previously, especially how PGC1 alpha, which is a transcription factor or transcriptional regulator,
yeah.
can reprogram muscle, and so he was approaching it through that lens. But that was really my first introduction to that really fascinating field.
Yeah, and he had this irisin uh, protein, which they have evidence is released from muscle cells in response to exercise. It gets into the brain and induces the production of a nerve cell growth factor, neurotrophic factor, BDNF, which is very important in in all sorts of things in the brain. Um,
Right.
okay, so then you mentioned that you were really interested in discovering new uh, bioactive molecules that have some relevance to regulating metabolism. And uh, there are many more molecules probably released or that increase in the blood in response to exercise than are known. In other words, a few are known, but there's many more that aren't known. So how do you go about identifying a new molecule?
I think that question of what you raised was the thing that kept me up at night as we sort of embarked on the study of exercise, which is that if you look at the field and you look at what's been done ever since, the term myokine was originally coined, IL-6 was sort of the original myokine almost, this is like 20, 30 years ago now. If you look at it as a field, there's just no agreement in the field. Everyone seems to propose a different molecule. People can't even agree on like what exercise is. And in fact, even if you look at CDC recommendations, it's sort of like any activity that increases your heart rate for 150 minutes a week that constitutes exercise. Are we supposed to think that walking around the block is the same thing as training for a marathon, is the same thing as, doing CrossFit and lifting weights? I mean, so it was that diversity of stimuli that initially seemed unwieldy, that we would have a particular answer that might not even be a reasonable answer, would only be a very narrow solution. There didn't seem that there could be sort of a unifying idea. And so that those concerns sort of kept me up a lot before as we embarked on this. But actually, those concerns were also relatively quickly dissipated when we first started to do experiments with a focus on sprint exercise. And we focused on sprint exercise, not necessarily because this is like sort of the best for your health, but just from an experimental perspective, it's the easiest to control. It's short, it's acute. Basically, any animal, including a person, can sprint. And so you can do it reproducibly. You can collect samples before and after, with the only difference being a few minutes between the pre and the post. And so we started to look in blood before and after sprint exercise from basically any animal that moves. We looked at mice, we looked at humans, we even looked at race horses that are racing. And when we
John,
started,
I saw that in your paper, and I don't know if you can see this up here.
yes, I see that there.
Yeah, myself, my father, and that's my grandfather in the 1950s. We used to train and race harness horses, standardbreds, not thoroughbreds, but
Not thoroughbreds.
and yeah, they're amazing. Right, so you're looking at sprint exercise. So in that in those situations, the person is going to kind of going beyond this threshold where you get a lot of lactate
Right,
accumulated.
right,
And but these horses are amazing because like typically a race is whatever, 2 minutes, and they're essentially sprinting for 2 minutes.
right,
And
right.
they're they breathe. I mean, if what one thing I noticed, I used to do a lot of distance running, right, where you're not really but these horses, particularly if it's warm outside, they'll keep puffing for 20 minutes.
Yeah.
After the even 30 minutes, they're still puffing. Whereas, like humans, I guess you don't push yourself to a point where you have to do that.
Yeah, I think that's a great point. I mean, there's sort of a few very interesting points here. I guess the first is that sort of coming back to our original search, when we looked at sprint exercise and we started to apply our biochemical profiling technologies to blood of all these animals before and after sprint exercise, it turns out that it's not that you get different answers depending on where you look. You get the same answer no matter where you look. And all these organisms induce the same molecule that we could detect in the blood. And that molecule is this lactate-derived metabolite, Lac-Phe. So the first thing was that, well, you actually have a very conserved, ancient pathway
Yeah.
that is conserved in sprinting across all these animals. The second point here relates to the race horses. I had been interested in sort of respiration and mitochondrial metabolism, energy expenditure during my time in Bruce's lab. At that time, studying brown fat, which was unrelated, but
Ah,
got me interested in metabolism as a general topic. And it was at that time that I learned that race horses are a form of extreme exercise because they actually have almost a 50-fold increase in respiration during the middle of their race, which is more than has ever been observed for any other mammal. So, for example, the best cross-country skiers, humans, will only increase their respiration and oxygen intake by about sixfold. Okay, so racehorses are just totally off the charts in terms of what they can actually do, how much oxygen debt they're actually accumulating in the middle of a race. And so that's why we chose them in terms of all the different systems that we could profile, because they represented a very extreme form of
yeah.
exercise. So that'd be interesting to look at them. And then just the third and last point, sort of anecdotally, is I'd always known about the race horses, but I didn't know whether we'd actually get a chance to study them. These are not sort of your typical standard laboratory animal. But what ended up happening was, here I'm based here at Stanford in the San Francisco Bay Area. Stanford, of course, is on the peninsula side, but across the bay used to be a racetrack called Golden Gate Fields. And when I called them and I said, I'm a scientist at Stanford, we study metabolism, we'd really be interested to see if we can get some horse blood. And they said, well, actually, it turns out that's not a problem at all because they sample the horses before and after a race all the time for drug testing.
Yeah.
So the A samples get sent out to the drug testing labs and they have lots and lots of B and C samples just sitting in the freezer. So I said, well, send those B and C samples over here and we'll put them on our instruments and we'll take a look. And that's the story of how we got the
Yeah,
racehorses.
that's cool. That's really cool. Okay, so okay, so then, but so you used mass spec to identify this. So you got lactate, which it's been known for a long time that when you do the sprint exercise, that accumulates. It's also been known that it can be an energy source for cells. There's specific transporters that transport it into cells. And then you have an amino acid. So how do they come together? There must be some sort of a reaction or enzyme to.
Right. So the molecule that we had found in the blood that was consistently increased and the most increased by like tenfold in all these organisms was this molecule Lac-Phe, which is a chemical conjugate of lactate and the amino acid phenylalanine. So, and that's how it derives its name. And what I would just say about this, sort of at the top, for because I understand most people are not chemists, so they think, oh, you're taking lactate and phenylalanine, you're conjugating them together. How do I think about that? And the way that we start to think about these types of molecules like Lac-Phe is that we call them liminal molecules. Okay. And so liminal is sort of an unusual word. In science, people want to say luminal. Okay, in sort of in the middle of a lumen. This is not luminal, this is liminal. And liminal means at the interface. And it's a word that's used to describe creatures like centaurs. So centaurs are liminal creatures because they're fusions of half human and half horse. But of course, a centaur is, you don't think about it as either a human nor a horse.
Yeah.
It is a liminal creature in its own right that has its own characteristics that does its own thing. And in the same way, this Lac-Phe molecule, the way that we have come to think about this is that yes, it is half lactate, and yes, it's half phenylalanine, the amino acid. But in fact, when you put it together, it's a centaur. It's neither lactate, it's neither amino acid, but its own liminal metabolite species. And sort of supporting this is the fact that we've identified the biochemical pathway by which Lac-Phe is made. It turns out there's a dedicated enzyme called CNDP2. This was an enzyme that was originally identified by my colleague Robert Jansen, almost 9 years ago now, but that reaction had only been demonstrated in vitro that CNDP2 could conjugate lactate and phenylalanine to make Lac-Phe. What we were able to show was that that's a real biochemical reaction in animals, because what happens is you could engineer animals to lack that enzyme. Okay, so they're genetically engineered, and when they don't have that enzyme, they can't produce Lac-Phe. And so that was the first demonstration in an animal that Lac-Phe production after exercise is due to a dedicated enzymatic pathway and biochemical pathway. And second, it provided an incredible tool because now we had these animals that could not produce Lac-Phe after exercise. And then the question becomes, well, what's wrong with these mice that can't produce Lac-Phe? And it was through that type of those series of questions that we then came to understand that what Lac-Phe does is it's produced after exercise, goes to your brain, and works on feeding centers to suppress appetite. And we think that the connection then to sprint exercise has everything to do with the fact that when people undergo, for example, high intensity interval training or so-called HIIT training, you typically feel terrible afterwards because you just had this really intense training. And we think that at least part of that feeling terrible and part of that not wanting to eat after very, very intense workouts relates to the production of this Lac-Phe metabolite.
And do all cells produce this in response to exercise, just muscle or other cell types?
Right. So the initial idea, because we are coming at this idea of myokines or muscle secreted factors, we thought that, well, lactate is produced from muscle. This Lac-Phe metabolite must be produced from muscle as well. But the surprise was that we knew the machinery that produced this metabolite, Lac-Phe, was CNDP2. And if you actually look at where CNDP2 is expressed, it's not expressed in muscle at all. In fact, it's most highly expressed in the gut and in kidney epithelial cells and in macrophages. It's not at all expressed in muscle. And then what we were able to do is sort of a genetic trick called conditional deletion, where instead of removing the CNDP2 from the whole animal, we could specifically remove it from different cell types or organs. And the conclusion from those experiments was actually that most of the Lac-Phe actually comes from the gut. It's gut epithelial CNDP2 cells that produce Lac-Phe because in the gut, if we specifically remove CNDP2 from the gut epithelium, those animals can't produce Lac-Phe either.
So,
And so very
so maybe that,
surprising.
so what's that, going back to what you were saying before about, so you've shown it suppresses appetite. So maybe that makes, pretend you're the gut, right? And you're in an individual that's just expended tremendous effort, and then you don't want that individual to eat food because the gut isn't really ready maybe to properly metabolize that food. I don't know.
Yeah, I think there's a that type of thinking, what you just said, connects to a few different ideas. I think that the first idea is sort of the suppression of feeding after exercise, and what's the logic there? I think this comes back to sort of the autonomic nervous system response and the whole fight or flight idea.
Ah,
Currently we have high intensity interval training, okay, but we're like in a civilized society where we're doing this indoors. Before we had hits, we were running away from predators. And in the running away from predators, what you have is a very, very classical physiologic response of your autonomic nervous system shutting down digestive
yeah.
pathways so you can fuel your glucose, you can fuel your running with glucose directed to your muscles, and you can sprint away and survive. So one way to think about what Lac-Phe is doing is that it functions as a chemical signal that parallels that autonomic nervous system response. So not only are you shutting down digestion through your nervous system, but you're also shutting down feelings of hunger with this chemical signal in your brain. Okay, so that's the first thing. The second aspect of this relates to what you're talking about with what is up with the gut and how come the gut is involved at all. Because what we're talking about here is that in exercise, you have a three-way crosstalk between muscle, gut, and brain. What's happening is the muscle is producing a lot of lactate. That lactate is being taken up in the gut where it's converted to Lac-Phe. And then that Lac-Phe is going up to the brain to suppress feeding. This seems like a very strange and complicated multi-organ circuit that we're talking about. Okay. What I would say here is that I would say two aspects about that. The first is that gut-brain communication is a big deal.
Yeah.
And there's a lot of other hormones that regulate feeding that are directly coming from your gut,
Yeah.
including, for example, GLP-1
Yeah.
and CCK and PY. Many, many gut hormones are. And so the idea that the gut is a source of appetite regulating hormones is not surprising. And so that's what we're really talking about here, that instead, in addition to all these peptides, that there's also a metabolite, a small molecule that's produced by the gut that can mediate gut-brain communication. That's the first thing. The second thing is, well, what's up with this whole muscle gut brain thing then? That first bit, muscle gut. And what I would say there is that, we originally found this pathway in the context of exercise where the muscle is producing the lactate. But actually later on, there was a paper that we did that was published back to back with another paper from Lydia Lynch's lab, where Barry Scott, who was at that time in her lab, showed that the Lac-Phe pathway could also be activated by dietary stressors, including, for example, metformin or even glucose. So as you consume carbohydrates, glycolysis in the gut can also produce a lot of Lac-Phe. In the context of eating metformin, which is a very widely prescribed anti-diabetic drug, that's inhibiting mitochondrial respiration in the gut and then also stimulating glycolysis that way. And so the sort of overall idea that comes from that is that this is a metabolite that's produced in response to glycolytic stimuli, and that, maybe sort of the primordial origin of it was in gut-brain communication in response to carbohydrates, that then was later co-opted maybe by sort of an exercise perturbation because you're feeding in lactate now. And so sort of the original genesis of this whole system was in classical gut brain, and then later on the muscle gut pathway was built in. So those are just some ideas for how we think about that now.
All right. So Lac-Phe produced in response to exercise in the gut goes to the brain to the hypothalamus, I assume, and inhibits hunger. And
Right.
where is it acting to inhibit hunger?
Right. So that's a question that we're still looking at now. We have generally in sort of unpublished work, we think it's hypothalamic, which is obviously an important feeding regulating center in the brain. And we've identified some neurons in that region of the brain, and we've also identified some sort of the downstream pathways. And so we're still trying to work that up. It's
Right.
still early days, but we think that it's going to be in that sort of that same feeding area that many other appetite regulating hormones talk to, including leptin and ghrelin, for example.
So will this centaur molecule, is it, does the lactate transporter or the so-called MCTs, monocarboxylic acid
Yeah.
transporters that transport ketones as well?
Right.
Does do they take Lac-Phe up into the cell?
So the answer is yes and no.
Okay.
But let me explain. So classically, so the monocarboxylate transporter, as you mentioned, is a very large family of transporters that move these monocarboxylates inside and outside of a cell. The classical ones that people talk about in terms of lactate and ketone transport are MCT1 and MCT4. And then sometimes they talk about MCT2 and 3 as well. So it's MCT1 to 4. What I can tell you is that those MCT1, 2, 3, 4 do not transport Lac-Phe.
Okay.
But we've actually identified another member, an orphan member of the MCT family
Huh.
that was not previously studied that in fact does transport Lac-Phe. And that is work that's unpublished, and we're sort of in the process of writing it up now. But what this is a really interesting story because what we actually show is that in fact the monocarboxylate there is a monocarboxylate transporter family member that's critical for mediating the transport of Lac-Phe from its intracellular site of production in the gut to the blood. And so when you genetically delete that monocarboxylate transporter from a mouse, and that mouse cannot get the Lac-Phe from the gut into the blood,
Ah,
it looks just like the mouse that can't produce Lac-Phe. So it's a fascinating system with interesting regulation, and it comes back to the idea that Lac-Phe is a centaur. So all those things that you thought about lactate metabolism, MCT1 and 4, those are not that's not how you think about Lac-Phe, but there's a related separate process that's going on for Lac-Phe.
so this Lac-Phe, in typical daily life of animals, including us that exercise, it it should be going up, and then coming down when we're resting, going up, down. In your studies, you also administered Lac-Phe to animals. Um, did you like compare continuous administration to kind of pulsatile that mimics more what would be normal? Because you might think there'd be differences in effects on metabolism.
Absolutely. So we have not done that experiment because our pharmacologic tools for controlling the dosing are not nearly that precise.
Okay. Yeah.
But you're totally right that the pattern, not only the magnitude, but also the pattern of change is very important here. And these are questions where we just need sort of better pharmacological tools to control that before we can answer that question. So what we can do for now is we can just give it as sort of a bolus. Okay, and when we give it as a bolus, we see that the animals have a suppression of their feeding behaviors without changes to, for example, their ambulatory activity or the amount of carbon dioxide they're producing or oxygen they're taking up. But we can't do sort of the more refined experiments as the one that you're describing here.
Yeah. So I've had a lot of interest in, and we had some evidence for a lot of labs actually that like with intermittent fasting or intermittent exercise, that they may be so good for health because you're getting kind of this biphasic thing where when you give the metabolic stress challenge, whether it's exercise or fasting, cells upregulate genes that are involved in cell stress resistance, conserve resources, autophagy, for example, or upregulation of antioxidant enzymes. And then during the recovery period, oh, and then mTOR is down, so they're not synthesizing a lot of new protein. Then during the recovery period, resting, eating, um, cells can go into a growth and plasticity mode, and the switching back and forth may optimize health. So,
I have no doubt about that kind of idea. And the in my opinion, the evidence supporting that there's really this idea of sort of the two faces of exercise. Because, for example, it's well known and well reported that after acute stimulus of exercise, for example, people running marathons or even hard workouts, you have this very, very powerful inflammatory response that kicks in right afterwards. Okay. And yet, in the long term, with sort of regular activity, you actually have a generally sort of anti-inflammatory environment. And so you already get a sense here that, it's not nearly so black and white physical activity as a stimulus, that everything has to do with sort of the pattern of the stimulus and the recovery, and somehow switching between stimulus and recovery is an important part of how the benefits are conferred. And I would also ascribe to that kind of idea in the context of Lac-Phe, because what I described to you was the sort of salutary effect from pulsatile
Yeah.
Lac-Phe administration or exercise bounce. There's some evidence now that actually in obese individuals, they have elevated, chronically elevated Lac-Phe. And so again, there's sort of there's a little bit of a yin and yang, a little bit of Janus type two-faced elements of Lac-Phe, where it seems that it's going to be important, the duration and magnitude and pattern of the exposure to this molecule. And
Maybe
we're
there,
still in early days.
maybe there's, so insulin resistance, leptin resistance, people with obesity, that's very common. So that the system to respond to these hormones is impaired through whatever, oxidative stress or whatever. And so the organs that are producing these hormones try to pump out more. I wonder if you could have something like Lac-Phe resistance.
Right. No, I mean you're certainly right that you have hyperinsulinemia in diabetes,
Yeah,
which would be counterintuitive given the glucose lowering effects of
yeah,
insulin. Absolutely, there could be something like that. I mean, we just started this whole area a few years ago. These
yeah.
are now the critically open
And
questions.
then like leptin resistance, right? Leptin suppresses appetite. So if your brain isn't responding to the leptin, then you feel hungry all the time. So
Right.
maybe there's something like Lac-Phe. That's your next grant proposal. Yeah. I was going to say with the mTOR, okay, do you think, and you haven't really, there's a lot more you've done in these animals where you knock out the ability to produce the Lac-Phe,
Right.
right? And so what if you feed them an obesogenic diet or do you think all of the beneficial effects you're seeing on metabolism and the brain, which I'd like you to talk about too, are just due to suppression of appetite? For example, GLP-1, these peptides, there's a question of how much of their beneficial effect in obesity and diabetes is simply to do with suppressing appetite versus some other effect.
Right. So maybe let me answer your question in a few parts. The first is that we believe there are other effects of Lac-Phe in the brain beyond feeding alone. What we have described here is an exercise inducible, sort of glycolytic stimulus inducible circulating blood-borne molecule that talks into the brain. And so we've been looking at every sort of
I see.
behavior that you could imagine to ask these animals that can't produce Lac-Phe, do they have a phenotype in this behavior? And if so, does that mean that Lac-Phe is important in that process?
Oh,
And so we're sort of now sort of systematically going through the list and we're finding other interesting behaviors outside of feeding control. Okay.
okay.
And we're trying to understand what is exactly going on there. So that's sort of the
Okay.
first. The second aspect is our current focus. You asked the question about how much is it due to feeding and body weight, and how much is secondary to that, or how much is independent of those of that primary effect. And I would say that we're still just in early days of understanding this. In the context of GLP-1s, I mean, those drugs have been around for at least 10 years now. And it's only now that we're starting to ask, okay, what about the anti-inflammatory aspects? What about the neuroprotective aspects? Okay. So we're still very much in early
Yeah,
days.
actually, my lab at NIH. So there's a medicinal chemist in my lab, and he worked with Josephine Egan, who does a lot of work on diabetes, and they developed Exendin-4, which is one of the first ones. And we looked at direct effects of GLP-1 and Exendin-4 on cultured hippocampal neurons, which my lab did a lot about. We did a little bit in mice, and they definitely these peptides have. It's an interesting story, but their receptors are coupled to a second messenger called cyclic AMP. It's probably the first one discovered actually a long time ago. It had to do with metabolism in the liver, I think.
Yep.
And and it's well known that cyclic AMP is very important for signaling in learning and memory. A lot of the stimuli that enhance learning and memory signal through that. So
Yeah.
yeah.
And I mean, we're actually, doing sort of analogous experiments now where we're doing whole cell patch clamp electrophysiology on different neural populations. We can do sort of whole brain wide map. I mean, these are very standard techniques, right? Brain wide mapping, where you look at what neurons are turned on when the mice are exposed to exercise or to Lac-Phe.
Nice.
And we're seeing interesting populations of neurons outside of the, both in the hypothalamus and also outside of the hypothalamus. And so this is sort of a growing area.
Yeah, that's very exciting. I'll look forward to seeing that come out in the next number of years. These are these are actually very time-consuming experiments, particularly electrophysiology, but other things too. A lot of work, takes a lot of person power to do this work,
Right.
and oftentimes collaboration with other labs.
Well, collaboration for us has been really key. I mean, hand in hand with us has been Dr. Yang Shu's lab down at Baylor College of Medicine. Yang was trained originally as a neuroendocrinologist, working sort of on leptin signaling in the hypothalamus with Joel Elmquist down at UT Southwestern. And so, this has really been sort of a partnership where we come in more from a biochemistry and peripheral metabolism perspective. And of course, we provide expertise in those areas. And then he comes in more from a neuroendocrine perspective. And it's been very fruitful because there's been a lot of interesting exchange of ideas in these two areas that should be related, but really there's been a surprising lack of crosstalk between the two communities.
Yeah, it's really good for postdocs and graduate students, people working in the lab too, to have that interlab collaboration because it enhances their scope of the techniques they're exposed to and the models and so on. I think it's really good training experience. Okay, let's let's briefly talk about fasting,
Yep.
which is so my own personal experience for my own health and my mental health and cognition. And I do both exercise and intermittent fasting. I think the exercise is more powerful, at least for me, in improving my mood anyway. I don't know about longevity. But so when you fast for an extended time period, the ketones go up in the blood, beta hydroxybutyrates, 1-acetoacetate, and they can be used as an energy source for cells. That's been known for a long time. And there's a lot of evidence that particularly beta hydroxybutyrate, BHB, has signaling function. That is, it's not just an energy source for cells. It can affect the expression of genes, turn on certain genes. We found it turns on the BDNF gene. And a group up at NYU, Moses Chow, his lab found that at the same time using different models. So, okay, so you talked about exercise, lactate goes up with exercise. You identified Lac-Phe, and I guess maybe other lactate amino acid conjugates. So then, did you decide then, okay, what about ketones and kind of take the same approach to that?
Right. So that's basically it. But I just want to add one more sort of bit of context here,
Okay.
which is that if we just sort of take a step back, most people, I mean, in the longevity field, and especially sort of the biohacking community, there's a lot of talk
Right.
about, exercise and intermittent fasting and
Yeah.
starvation, and these two types of protocols.
Yeah.
But they're very different. I mean, there should be nothing related about these two things. Okay, one, you're moving your body, you're talking about, carbohydrate metabolism and lactate. In the other one, you're talking about not eating in fasting and intermittent fasting or time restricted feeding or starvation or ketosis. You're talking about not eating carbohydrates, ketogenic diets. And you think how can these two things be the same? They're not. Okay. And one thing I'm moving and one thing I'm not eating, they should be unrelated. And in many, many aspects, they are unrelated. Okay. Our body has fundamentally different fuel sources for these two things. In the context of exercise, we're talking about glucose catabolism.
Yeah.
In the context of ketosis and fasting, we're talking about lipid metabolism because we're burning our fats. Okay. And yet, what really stuck out to me as a chemist was that despite all of these differences, the fact that they're stimulated by different things, by moving or not eating, the fact that the fundamental fuels are different, glucose or fats. What nature has decided to do, for reasons that are beyond my intelligence, is that the downstream critical molecule that is produced from these are chemical cousins, lactate and beta hydroxybutyrate. If you actually go into a textbook and look at the structure of these two molecules, you would get them confused.
Yeah.
They're both hydroxy fatty acids. The only difference is that lactate is a C3 and beta hydroxybutyrate is a C4. So the only difference between these two things is a single carbon group. Now, why nature has decided to make chemical cousins out of lactate and beta hydroxybutyrate when everything else about their physiology is so different, I have no clue. But that is what has happened, and that is what the biology is.
I have an idea. I have an idea.
Yes, I would love to hear your idea about this.
Well, it's the evolutionary perspective that food scarcity is a major driver for evolution, including maybe particularly brain evolution. The need to get food and compete for resources, avoid predators. And to acquire food when food is scarce, you have to expend a lot of physical effort, whether it's, chasing down a prey animal, running whatever mile and killing it, or foraging. And so one possibility is that both of these molecules evolved to support the brain under those conditions where you're both deprived of food and expending energy at the same time.
That's a really interesting idea. I hadn't heard about that type of idea before.
I wrote
So,
that in, I put that, my first chapter in my intermittent fasting revolution book is on, intermittent fasting revolution book is on evolution.
got it.
Yeah.
I've always been, I think chemically it's quite fascinating,
Yeah.
this idea of sort of, chemical cousins, lactate and ketones. Now, what I just described to you was that lactate can be derivatized into a lactate metabolite, Lac-Phe. And so, sort of by extension and extrapolation, we wondered, well, couldn't the same thing happen to ketones as well? Now, the reason that that idea was so unusual at the time is exactly what you talked about, because during fasting and ketosis, there had only been two biochemical processes described. There was the production of ketone bodies called ketogenesis, and then the catabolism of those ketone bodies for energy, so-called ketone oxidation. And ketogenesis and ketone oxidation was thought to be the totality of it.
Yeah.
But what we were talking about here is that if the same thing that we just described for lactate could apply for ketones, then what you have is not two pathways of ketone metabolism, but three. Because in addition to ketone body production and ketone body catabolism, you also have a shunt pathway of ketones, this third pathway that produces ketone metabolites. So at the time, it was sort of a very unconventional idea because, people have been studying ketone biochemistry for, decades.
Yeah.
And every textbook you ever read says, there's only these two pathways.
Yeah.
And so we decided to test the idea because we had such compelling rationale from what we just discovered about lactate. And we had sort of compelling chemical similarity between lactate and ketones. And so what we eventually discovered then was that, in fact, yes, there is the third pathway of ketone bodies, that beta hydroxybutyrate can be derivatized by amino acid conjugation to produce these ketone metabolites like BHB-Phe, that in fact, the biochemical machinery that does that is exactly the same as what I just told you for lactate. It's the same CNDP2 enzyme. And so what this enzyme really is a sensor of how much lactate or how much ketones you actually have. And then depending on how much it's measuring, it produces a corresponding amount of lactate metabolite or ketone metabolite.
Are there any human disorders that are caused by mutations in that enzyme?
Not yet that we found,
Okay.
but we're starting to look more carefully.
Well, it may be strongly selected against during evolution. It seems really important. Okay,
Right.
so what did you find then when did you do fasting? Okay, so you've got these uh, what is it, CDNP2
Yeah, CNDP2. That's
knockout mice, and then you fasted them or you put them on ketogenic diet or something like that?
correct.
Yeah.
Correct. So the thing about these animals to know is that actually they're pretty normal. Okay, the mice that lack this enzyme, they are born in the proper Mendelian ratios, they grow and they develop normally. They even have normal body weight if you just give sort of, if they're sedentary in their home cage. Where the phenotypes are revealed is where you apply the right stimulus. And you have these very, very important but poorly controlled generally, so-called gene by environment interactions, these G by E interactions. Okay. So what we found, so the way to think about that in the original context of our exercise discussion is exercise is a glycolytic stimulus. And so there's an important glycolytic stimulus by CNDP2 genotype interaction,
Yeah.
where by specifically provoking the lactate part of that pathway, you reveal this difference in body weight in those animals. With ketosis perturbations like ketogenic diet or exogenous ketone ester, we revealed the ketosis by CNDP2 genotype interaction. Where now, for example, if you give ketone esters to a wild type, I mean, these are the things you can buy from delta G or HbMN ketone, you can actually see effects of those ketone esters on body weight in mice, but the CNDP2 knockout mice are resistant to that effect of exogenous ketones. And similarly, they're resistant to the effect of ketogenic diet. And we can show that those are specifically perturbing the ketone amino acid conjugates rather than the lactate derivatives.
The ketone ester actually was developed by a colleague of mine at NIH a long time ago, Richard Veach. And then he collaborated with my lab. We showed if we put it in the food of mouse model of Alzheimer's disease, it had beneficial effects. And then he collaborated with Kieran Clark at Oxford to do a lot of exercise stuff. It was pretty interesting, both in initially rats, but then in human elite British cyclists who
Right.
who before they started taking ketone. So there was a number of years where the British cyclists were taking the ketone ester and no one else was. And prior to then, only one British cyclist had ever won the Tour de France. And then during those years, there was like 5 years. They, well, now they went through the anti-doping thing. It's an energy source.
Right.
It's like taking glucose, so it's not a big deal.
I mean, in the endurance sports world, the ketone loading is now becoming a big deal.
Yeah,
It used to be carb loading.
yeah,
But especially as you start to think about sort of prolonged exercise where you're using lipids and ketones now for fuel, there's sort of a growing effort to understand the effect of exogenous ketone supplementation. So it's a very fascinating area. The other aspect that's interesting from a brain perspective is that, the connection between ketosis and sort of brain health is very strong in multiple
yeah,
dimensions, right? First of all, that clinically, if you have drug-resistant seizures in children,
yeah.
you put them on ketogenic diets or other starvation diets, and that, I mean, in some of these kids, it just brings down their seizures to like nothing. And so, and there's many other indications of sort of ketosis in terms of mood and cognition and many other aspects of brain health. And so, in sort of the same way that we described for exercise and for Lac-Phe, this sort of ketone shunt and these BHB amino acid metabolites now gives you sort of a new lens to reinvestigate how much the effects of intermittent fasting are actually due to BHB itself, ketogenesis itself, ketone oxidation in the brain, or maybe due to this ketone shunt. So, for example, one of the questions that we're wondering now is because epilepsy is sort of the strongest clinical indication associated with ketosis,
Yeah.
we're now looking at mouse models of epilepsy and trying to understand as you start to cross in CNDP2 knockout
Huh.
mice into these
Yeah.
mouse models, is there any modulation of the epilepsy phenotype in the convulsion phenotype? And if so, then you could ascribe that to a specific function of this ketone shunt potentially, rather than some other aspect of ketone body metabolism. So that's sort of like one of the things that we're trying to do. But there's many other aspects. I mean, even beyond the brain, the ketogenic diets and fasting have been implicated, for example, in cancer and many other sort of areas. And
Yeah.
so the way that we think about this is that before you would have only investigated these through two lenses, basically ketone body production and then catabolism. And now what you have is an opportunity to reinvestigate all of these phenomena through three lenses, including this ketone body shunt.
Yeah, it's very exciting from a translation, from basic science and translational science standpoint. Um, in the future, these ketone esters are still kind of expensive for people to be taking, average person to be taking very often.
Well, they also don't taste very good.
No, but there's some companies like there's one that I consulted for, a little Kinetic, whose theirs tastes pretty good. But I just do the intermittent fasting and hope that's just as good and save money. But uh, okay, what about translation? I know you need to leave pretty quickly here.
Right.
Is there anything on the horizon? You probably patented these chemicals, and what's kind of your vision without giving any secrets away, I guess.
No, no, I'm happy to talk about
Yeah.
it. So I think there's a few different directions. On the basic science front, right now we still don't understand the direct targets of any of these molecules in the
Yeah.
brain. We don't have receptor molecules, we don't have direct interactions.
Yeah, yeah.
And until we find those, we won't be able to really sort of from an FDA type of perspective go through and develop, chemical matter into potent receptor modulators that could target some of these pathways. What I can say about that from the basic science side is that, we've obviously explored the obvious candidates. I can tell you that we don't believe this is a G protein coupled receptor. We think that we're more in ion channel land,
Huh.
and that search is still ongoing.
Interesting.
The other aspect of it relates to the idea of sort of uh, relates to the idea of these ketone esters, because there's a possibility that maybe for example, some of these ketone amino acid conjugates are potentially more useful in terms of direct dietary supplementation. We don't have evidence for that, but that's one potential idea. And in fact, there's a company that is sort of pursuing that idea now and has licensed some of those materials from the perspective of developing a supplement plus. And so those are sort of the active translational aspects. Obviously, my lab as a basic science lab is fundamentally focused
Yeah.
on sort of basic molecular mechanisms of these pathways, and so we're really heavily invested on that first part. But those are the
You know, you mentioned the ion channels with so glutamate's the major cytochrome transmitter in the brain. There's two receptors. One of them, the NMDA receptor, is a calcium channel, and there's drugs that block kind of not competitively, but they get in the channel. Uh, Phencyclidine is one. Another one's called MK-01, and they're kind of lipophilic, right? And so maybe these molecules are actually getting in the channel pore.
Okay, so that was a very, very prescient insight. So what I can tell you is our thinking about how sort of the chemical logic of this whole thing and why, for example, phenylalanine. We know that when you conjugate phenylalanine to either lactate or ketones, the whole thing becomes very, very greasy and lipophilic. And it's much more lipid-like than you would expect just by looking at the structure. Okay. We think that that aspect of it is a fundamental switch in terms of explaining what's going on. That you take something that's entirely water soluble, either ketones or lactate, and you turn it into a lipid. Now, the other aspect of it is that in addition to what you just mentioned with the NMDA receptor, there are many, many ion channels that have bona fide and complex interactions with lipids.
Yeah.
And so we're really thinking about not just the orthosteric interactions that you're talking about, but complex allosteric interactions outside of the orthosteric binding pocket across a diversity of channels that may be mediating some of these effects.
Okay. This is great. We could go on forever, John, talking back and forth on this, but kind of the key point is you've identified two previously unknown biochemicals, one produced strongly in response to exercise, the other, I guess, fasting. It goes
Yeah.
up with BHB, and they have very important effects on metabolic health and apparently brain plasticity too. And this opens up a tremendous amount of future research. We just my back and forth with you, right? We touched on all sorts of things that can be done now to extend this. So I appreciate you taking the time. I know you're really busy, and I hope things go well in these troubling times where we have not too thoughtful individuals controlling the money that goes to research and not recognizing that our country was built on science and then technology that was developed based on the science. And, you know, without the engine of science, technology is going to falter or slow down a lot, and it's not going to be good. I,
Right. Well, Mark, thanks
I'd
for
like
having
to
me
say
on.
one other thing there, that there's countries that the administration, thinks are our enemy, essentially. China, for example, they're not our enemy. I mean, we all know scientists, right? They're Chinese. But it used to be that if I had a postdoc, say in the 1990s or even 2000, early 2000s, come from China, outstanding, very smart individuals, highly productive, and a lot of them going to get faculty positions here back then. But now, within the last 10 years, I've had postdocs come to my lab at NIH from China and then go back because they got better lab setup, better funding situation there. So we're going to get behind on science and it's not going to be good. We got to think far into the future, not in the next 4 years. And these things, there's a big lag between the basic science and realization of its benefits for society.
I totally agree with that sentiment, and I think you know the I think from the perspective of a long-term strategy, we really need to be cognizant that these small investments now pay off
Yeah,
in sort of an outsized way. And I think from the perspective of a university, for example, here at Stanford, that it's partly our responsibility as scientists working at these places to maybe to better explain,
yeah,
or not just that, but also to sort of properly engage in a long-term relationship with not only Congress, but just like Americans generally.
yeah,
And it's through strengthening that understanding and relationship can we really ensure that everyone is winning at the end of the day.
yeah. I agree 100%. Well, thanks again. I'll let you get back to the lab and your lab members interacting with them.
All right. Thanks so much, Mark.
Yeah. Work hard and have fun. That's my motto.